All right, here we go. Cool.
All right, you can see my screen.
Mm-hmm.
Okay, so this is so I'm going over the AMC press
Yep.
prepping with like the Jack Weston plugin so that I get like
Yep.
this. Yeah.
So
Yeah.
okay, so this is what we're gonna do. So yeah, I'm going to have you like read out loud like slowly. Now we're gonna look for something called buzzwords, which are essentially words that, well, they'll likely ask you questions on. Um, and oh yeah, you could do that.
Okay. So you want me to just start reading them?
Yeah, yeah. And then like I'll kind of like interject and ask you things.
Okay.
But yep.
Do you also want me to like highlight as well, or just stick to
Oh
reading?
no, not yet. Yeah.
Okay. So human immunodeficiency virus type 1, HIV-1, integrase IN. It's a viral enzyme that mediates the integration of viral DNA into the host chromosome.
Okay.
Um,
So so a lot of passages, especially in BB, will be about like an enzyme. A lot of times they'll be like they'll introduce some enzyme and they'll talk about like the enzyme's active site, and they'll usually say like the active site features like certain amino acids, and that will make you think like, okay, what type of amino acids do we have, blah blah blah. And a lot of times they'll end up doing some type of experiment where they're going to make mutant variants of those amino acids. Like usually they'll do something like let's say we have like aspartic acid at the 39th position, they'll replace it with like alanine, which is like, just like the amino acid with like a methyl that doesn't really do anything. So a lot of times you'll be losing like the function here, and so they'll have some type of like kinetic data or something like that. A lot of times they'll be talking about something like that. So
yeah.
and we see that right here with the enzyme. So get ready, just to be doing a lot of stuff on enzymes. Yeah.
Okay. Yeah, and I guess like keywords that are like sticking out to me, viral DNA. So right now I'm kind of like orienting myself into like, okay, everything I know about the virus, how it
Mm-hmm.
influences like
Mm-hmm.
host and whatnot. So I definitely like highlight all of these. I guess my habit is that I would just like go ahead and like highlight all the abbreviations for things. I don't know if that's like a waste of my time or
It
uh,
depends. We can see like, yeah, I guess we can see how that goes. So yeah.
sure. And I think another thing that I would do is like it mentions the host chromosome. So like I would probably highlight that part just to kind of like orient myself in host chromosome 2.
Yeah,
Um,
so what are you like visualizing as you read this?
I mean, I'm thinking of like lysogenic pathway, lytic pathway, like just transduction, transformation, like everything that has to do with like viral pathways. I'm just kind of like envisioning that. And
Okay,
so
okay.
I suppose like whenever they start throwing out like names and stuff like that, I think it's just like easy to get intimidated by it. And I think like I think I get overwhelmed with like I guess like the over like the I guess how much detail they kind of throw at you and
Mm-hmm.
not realizing like, okay, well, is that even relevant to like this
Mm-hmm.
type or not? And
Yeah.
so I think one of the ways that I've been trying to get past that is to just like start taking notes. But
Uh-huh.
I think it's kind of depending on what the passage is, it could end up being like a complete waste of my time, I've found in some passages. And so, but I think, I guess I'm trying to like find a way to like have a systematic way of just like approaching passages. And maybe it's just not appropriate to approach every passage the same way, I suppose. And so I guess maybe that's probably my thing to kind of figure out
Yeah.
which passages.
So I'll tell you what I'm visualizing, right? I'm
Mhm.
visualizing HIV having its own viral DNA, and the host chromosome, like the host cell, having its own DNA, and IN is like helping the HIV DNA get into the host DNA. Like I'm visualizing like literally like HIV as a circle, host as a circle, and then the IN
Oh,
helping that HIV DNA get into the host DNA.
okay.
So like yeah, like a little like just imagining like circles and like IN is like here you go, like go in, go in. So that can I mean like visualizing, is always something that well, it may not always be something that someone does, but it can be like an automatic thing that you can think of to help you like because you do want what you said was right about like what level of detail do we need to I guess like view things and starting off like in a simple way I think would be a good place to start.
Okay.
So just like yeah, I'm just imagining these like circles. I'm not worrying about anything too specific yet.
Okay. Fair enough.
Okay.
So going on to the passage, this enzyme IN 288. So I'd like highlight that because I would assume that they might ask about like amino acid.
Yeah.
Uh,
Okay. Okay. Tell me what they might ask about from that.
um, Usually when I've seen them, they'll ask like, okay, well, how many like kilodaltons are in
Yes.
like
So
X, Y?
what um must you know to answer something like that?
Oh, say it again.
Oh yeah. So like what you said, like what information do you have to know to figure out something like that, like the weight in like kilodaltons or something?
Yeah, so the conversion between like, so how many amino acids you have, in like, I think 1 kilodalton is like divided by 110 to be
So
like,
the 110 is definitely there. So one amino acid would be 110 Daltons,
okay, yeah.
right? So like that's that conversion factor to know. It's like on just the average, so you could use it for any amino acid, but like yeah, like the average weight of an amino acid will be 110 Daltons. So if they tell you 282 residues, just know that if you had to, you can calculate the weight of the protein in like Daltons or kilodaltons.
Okay. And 288 would be how many amino acids?
Mm-hmm.
Okay, gotcha.
So right there is a buzzword right there, the residue. So, and it's good that you recognize that.
Okay. So composed of 3 domains. So we have an N-terminal domain, catalytic core domain, CTD, and the C-terminal domain. So I'm kind of envisioning now like an amino acid essentially, N-terminus, C-terminus, and then everything in between.
Yeah, and particularly what would be like important for us to like out of the three domains here.
The let's see, is it the yeah core CCD domain
Yep,
as
because we
our
know that enzymes are like biological catalysts, right?
Mhm.
So like it's cool that there's three domains and all, but what we're really trying to think about is that the domain that actually makes the enzyme do whatever it does, right?
Right.
So it's kind of like what I mentioned like at the beginning, like a lot of these passages will be asking you like, okay, this is an enzyme and this is what it does, and at its active site, there's going to be certain amino acids. So yeah, good. Okay.
Yeah, and so in that domain, CDC,
Mhm.
CCD contains 3 highly conserved residues. So these amino acids at these particular positions
Yeah.
and
Do you,
mutation. No,
oh yeah, sorry.
no, no.
Do you recognize what those are, like from the single letter abbreviations?
Yeah, so these two are aspartic acid, and then this
Uh-huh.
is glutamic acid.
Good. So what type of, tell me what, tell me more about them. Like what properties would they have?
I know they're acidic, so they'll have the like if they were to ask me like what the charge is, these would all have -1 charges.
Yeah, good. And it kind of makes sense, right? Because, you know, for an enzyme to do stuff, like if it has a bunch of like negatively charged amino acids at its active site, then it's probably going to bind to things that have positively charged like amino acids. So it's usually going to be something special about the amino acid, so that can help. All right, good. And yeah, the mutation part.
I have a quick question for you, actually. So, like, when you mentioned the active site, like, that's
Yeah.
not something that immediately like came to my mind.
Mm-hmm.
So, what about like, because I think you mentioned that even like in the beginning of
Mm-hmm.
when I was
Yeah.
studying. So, what about this kind of told you that like, oh, okay, active site, I need to orient myself there.
Yep. Okay. So all proteins will have the N-terminal domain, C-terminal domain, right?
Mhm.
But for a protein that's an enzyme, it's gonna act as a catalyst, right?
Yeah.
So what part of it, acts as that catalyst? Um, it'll be some part that has what we call like an active site. I mean, here you know, they didn't call it that by name explicitly, but they tell us catalytic core domain, and we know that an enzyme is a biological catalyst because so essentially what we want to know is what makes an enzyme, right? Uh,
Okay.
so we know an enzyme is going to be a protein, but you could have proteins that aren't enzymes, right?
Mhm.
Like a structural protein, something like that, right? So what makes an enzyme protein like special, right? It'll be that it can have activity, like enzyme activity, right?
Yeah.
And that means that it'll have some location on the enzyme, on the protein, that will be able to take like a substrate and turn it into like a product. So, so like, let's say we have, again, I'm gonna draw like a circle. Like a lot of times, like it's, kind of like just, I guess, visualizing it in a simple way. Like, let's say
Mhm.
this is our enzyme. Um, what makes it an enzyme other than like being just a regular protein is that it's gonna have some site where, let's just say it's like this, where let's say a substrate can go in, bind to it, and then be turned into like a product.
Yeah.
Now that this is like going to be the active site here, which will have some special amino acids. And when I say special, I mean that's kind of another like way that I kind of go over amino acids because typically they just tell you like, okay, memorize them, like the list of polar, non-polar and stuff, but I think it's best to view them in like this. Like first, uh if they're charged, right? So that would be like the two acidic ones and the three basic ones. And then rather than writing polar, I'm going to write a partial charge. Because if you think about it, something that's polar really just has a partial charge, right? Like if you had like a carbonyl group, the oxygen is electronegative, so it pulls electron, density towards itself. So this dipole goes in this direction, which means that the carbonyl carbon here has a partial positive charge and the oxygen has a partial negative. So instead of just saying like this is polar, memorize it, and this is non-polar, memorize it, I want like, well, how I usually like teach the amino acids is like first the charged ones, because they're charged, they're going to have, properties that are going to be important. Um, and then what's the second most important thing? Well, if it's not charged, then if it's partially charged. So that would be essentially STY. And these are what can you tell me about these amino acids?
They all have hydroxyl groups.
Yeah. And on the test, the hydroxyl group is going to be your most common nucleophile.
Yeah.
So when I say nucleophile, a Lewis base, like an electron paired donor. So STY is going to be special in that kinases like will use them to phosphorylate things.
Mhm.
So STY could be these three, polar ones, partially charged. But also if you take the acidic ones like aspartic acid and glutamic acid, and in their side group, like they would have like a carboxylic acid as a R group, right?
Yeah.
But if you change that to this, what type of, you know, functional group is this now?
Amide.
Yeah. So if you take them and you turn them into amides, the aspartic acid turns into asparagine, and the glutamic acid turns into glutamine, and those will be the other two partially charged ones. So
Mhm.
that's 5 charged, 5 partially charged, and then the 10 that remain will just be the nonpolar ones. Um, but yeah, essentially, yeah, you want to look for like just things that make like an amino acid special. So, alanine is very non-special. So a lot of times they'll do like the mutation. I mean, here they tell us explicitly that a mutation doesn't matter if it turns into alanine, like a mutation at all will get rid of that ion activity. But
Mhm.
other passages won't tell you that explicitly, but they'll just say like, they'll say like D64A, like they'll probably put it in like a table or something. But yeah, so yeah. Did
Yeah, I
that
think
answer your
that's
question? I feel like I kind of went off into like a
yeah, it does. I think one of the things that I wasn't doing was like reminding myself that like, oh, enzymes also have like these properties that
mm-hmm,
include active sites and whatnot.
mm-hmm,
Yeah.
mm-hmm.
Something that like wasn't really triggered in my memory. Like it's something that I know, but I wasn't reminding myself when I would read this. So
Yeah. Yeah, the reason that I put it that way is because we know that enzymes are proteins, but what like what makes what makes an enzyme protein different from a regular protein? It'll have to have some active site.
yeah.
So,
Okay.
okay, cool.
Okay, so integration of vDNA into the host chromosome proceeds by two well-characterized steps, both of which are catalyzed by IN. In the host cell cytoplasm, I probably highlight that just to like orient myself. Um, host cell cytoplasm, each end of a newly synthesized vDNA segment is bound at a specific long terminal repeat, so LTR, nucleotide sequence by one ion dimer. Upon binding,
Oh,
ion
so
cat.
yeah. So tell me what's happening here, like in terms of like what they're saying.
Um, so each of the vDNA segments are bound at a like a specific, I guess, region, I suppose, or like a sequence. So it sounds like maybe I think the first thing that's like coming to my mind is like, oh, maybe telomeres or like something to that effect, like something similar. Maybe not
Yeah.
quite.
So tell me that part again. You were saying like a region of the vDNA, like tell me about like what region or how they describe it.
So
Oh,
so
like
each
yeah.
of these vDNAs, they have like segments um that are bound, I guess, to this particular like sequence of these repeats.
So when you say each of the vDNA,
Mhm.
tell me what you mean.
Okay, well, maybe not like separate vDNA, but like on each end of.
Each end, right? So okay. So all right, good. So yeah, each end of a newly synthesized vDNA segment is bound at a LTR. Um, and then yeah, you can continue that
Yeah.
the end of that sentence.
So they're bound by each um, at a specific LTR nucleotide sequence by one IN dimer.
Mm-hmm.
So a dimer is just kind of like it's like almost like twins, I suppose, kind of paired with one another. So
Mm-hmm.
that's what I
Yep.
think.
So okay, so the dimer, if they asked you the weight of it, you would be able to find it, right?
Okay, could they specifically mention that it was one dimer? Is that
Mm,
what you're pointing to, or?
yeah, so a dimer, tell me again.
Okay, well, yeah, so it's like two. So like
Mhm.
whatever the weight is, you just multiply by two type thing. Yeah.
Yep, yep. All right, cool. Now let's see. Um, so each end of that vDNA segment, right, has an LTR, which is going to bind to an IN dimer, right?
Mhm.
So, what are you visualizing?
Um, hmm. So I'm seeing like a long sequence being attached to like the N terminus and the C terminus of this enzyme. Um, and so I guess maybe so maybe the IN dimer is kind of like connected from like one C terminal and then the N terminal of another. And then
So
these
that
L.
would be for like a protein, right? Um, but what end, like when we talk about the ends, what are we talking about?
Yeah, I guess I'm not clear on that. Like the ends of the nucleotide sequence, is that what you're referring to?
Hmm. So where the ends are of the vDNA segment, right?
Mhm.
So, because like an N terminal, C terminal, that would be for like the protein, right?
Mhm.
But we're talking about the vDNA, right? So, so they're saying that each end of a vDNA segment has these LTRs that are going to be bound by an IN dimer. So I'm imagining vDNA, and at these ends, there's LTRs, but that's just what's in at the ends of the vDNA. But each end is going to be bound by IN, like an IN dimer.
Mhm.
So I'm visualizing like a segment of vDNA, and each of the ends are bound to an IN dimer. So I drew an IN in a circle and another IN in a circle. So does that kind of make sense from what they wrote?
Yeah, and I guess like I mean when I'm reading so fast, like
Yeah, yeah. And another thing is, yeah, you definitely want to be like it's obviously hard to easier said than done. Like obviously when you're taking the test, it's timed, you know. How are you going how are you gonna read slow, right? But that's why that's why like I think it's good to like read slowly, and as you read, like kind of, I guess, like visualize or kind of like think about what you're reading and like what it means like as you're reading it.
Mm.
Um because otherwise if you read it too quickly and you don't understand it, you'll have to reread, and you don't want that, right? You want to like measure twice, cut once, right? But yeah, continue what you're saying.
Um, what do you want me to continue reading or?
Oh, no, no, you were saying something, I interrupted.
Oh no, I mean I was just mentioning that like
Mhm.
if I were to have like if I wasn't timed,
Yeah,
obviously
yeah.
I think I would have been able to like just
Hmm.
have, I mean it is what it is. So,
Hmm.
but yeah, to your point, I suppose I should be kind of like actively reading, but
Hmm.
also not reading just for the sake of like just like
Yeah.
reading. So,
Hmm.
so when if you were going over a passage, would you be like taking notes or how do you usually?
Yeah, I would write certain things down. Like usually, um, I would write like relationships in terms of proportionalities.
Okay.
Um, but for a passage like this, yeah, I would draw what I just drew down. Because like because you want, like, visualizing is kind of a way to encapsulate like thoughts in a picture. Like if a picture is like worth 1,000 words, it's very efficient to imagine a picture.
Mhm.
So I'm reading it like each end of a newly synthesized vDNA segment um, is bound at an LTR by one IN dimer. Um, so I'm not going to be too focused on, I guess, the details of eDNA or the details of LTR. Like I didn't even really write LTR here,
Mhm.
because I see that it just is what's going to be at these ends. But what I'm really trying to see is just the ends of the vDNA segment that are bound by an IN dimer. So I just kind of drew it. So sometimes in this case, I will draw pictures and stuff, but in other types of passages, I would write proportionalities and things like that. But yeah, for this one, I would draw the pictures. So yeah, I would say to definitely try to, I guess, like practice like visualizing. Because if you can get to the point where you can kind of like, as you read it, you can kind of visualize it, that can really help. And it doesn't, it's not going to be like a very complex picture, but like a simple picture to just help you kind of understand something. Because if you write it with words, it's going to be just more stuff to read. So sometimes drawing works. Yeah.
Okay. So continuing on reading. So upon binding, ion catalyzes cleavage of a GT dinucleotide from each 3' vDNA terminus. Still bound to either end of the vDNA segment, the ion dimers join to form a tetramer. So now I'm picturing like almost like them connecting.
Hmm. Yeah, that's
Oh,
exactly, good job. You just visualized it.
yeah.
But yeah, you see how you got it, right? Now it's
Yeah,
coming together.
yeah. I think the like I don't think I would have been able to do that if I completely missed the fact that like, oh, it's oriented in this way, right? Like
Hmm.
because like if I completely missed that, then I'm just thinking like, oh, all of a sudden there's a tetramer somehow.
Hmm. Hmm. Yeah.
So, but okay.
Yeah,
Um,
and also like it's so you were able to visualize that without being too concerned about this cleavage of a GT dinucleotide. Because
mhm.
there could be stuff that is more complex that's not easy to visualize like that. But it's like you're kind of like, okay, I know I can look into that later. It's not the most important thing now. The most important thing is like that I'm visualizing them coming together. Yeah.
Okay. Yeah. Okay. So which then binds host cell protein LEDGF at the CCD. This vDNA IN LEDGF complex enters the nucleus where LED tethers it to host chromatin. Then ion catalyzes the insertion of the vDNA into the host genome. Okay. I think in my head, I'm just telling myself that like, okay, it somehow gets into the host genome. And then like, I'd probably just go back to like, if I have to refer to like the specific way that this complex is
Yeah,
entered.
yeah. I would like, so maybe I would draw like IN, 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 that tetramer, and then the vDNA coming together, like you said, right? So that's when they come together to make that tetramer. And then maybe I would draw LegF. Um, so maybe like I could draw like, okay, well, let's see. Host cell protein LegF. I'm just gonna draw another circle. And this can bind to that tetramer, right? Because they say the tetramer, which then binds host cell protein LegF at the CCD. So where's the CCD going to be? Is it going to be um part of the vDNA? Is it going to be on the LegF? Is it going to be, I mean, before anything binds, is it going to be on the IN?
It's gonna be in like the core, so like in the near the center-ish, or like
So
after.
would it be like in vDNA, LegF, or IN?
Oh, an ion, because it
Yeah,
has the terminal.
good, good, good. So okay, so all right, this LegF thing comes from the host cell. IN comes from the virus, and yeah, the LegF binds to the IN at the CCD, that active site or the catalytic core domain. And then this vDNA IN LegF complex, which is like this entire thing here, can then enter into the nucleus. So, maybe I'll do like this, or I don't know, like maybe I'd even write enter nucleus or something. Um, where the LegF tethers it to host chromatin. So maybe I would draw like, I don't know, like a bigger picture like this with the LegF attached to that, and then I guess maybe something like this to say that it can then enter or incorporate itself into the host DNA. Maybe I'll just put something like this to say host DNA. But yeah, I would kind of, yeah. So the way that I'm seeing this is like, okay, LegF from the host cell binds to that IN tetramer, and then it's like allowed to enter into that nucleus. Um, and once it's in that nucleus, LegF like tethers it to the host DNA or chromatin. Um, but yeah, pretty much that.
In the last paragraph, small molecule ion inhibitors show promise as antiviral drugs against HIV-1. One class of these inhibitors are legends, which bind ion and shift its oligomerization equilibrium toward an inactive tetramer that cannot bind vDNA.
Mm-hmm.
Another class of small molecule inhibitors are short length or let's just say ODNs, whose sequences mimic LTRs. ODNs are competitive inhibitors of vDNA for binding to IN. So I'd probably highlight competitive inhibition.
Mm-hmm. So okay, so we start this paragraph off by talking about enzyme inhibitors. So like that's, you want to think about those. And so one type is legin, um, which apparently would bind to the IN and make it inactive, right? So that's going to be some type of like whatever, uncompetitive, non-competitive, one of those other types of inhibitors, right?
Mhm.
So that's the legend. And then the ODN, they do say competitive inhibitors, but if they didn't, from this, you would, oh yeah.
Like classroom was the light was turning off. So
Okay. Yeah, so if they didn't say ODNs are competitive inhibitors, you would be able to kind of tell by them saying that the ODN sequences mimic LTRs, right?
yeah.
Because the substrate, so like IN is the enzyme, and the substrate is the vDNA, particularly the LTR part of the vDNA, right? So
Mhm.
if it looks like the substrate, that's going to be a competitive inhibitor because they compete for that active site, right?
Mhm.
So, okay, so we have ODNs that can be competitive inhibitors and legends, which can be a different type of inhibitor, right?
Yeah.
Okay, cool. So we pretty much have what we want or need. Um, how do you feel?
Yeah, I feel solid about my
Hmm.
rundown. I think we have enough. Yeah.
Cool. Okay, let's try some of these.
Okay. So what bond is cleaved by ion during the first reaction of integration? So my immediate thought process is to go back to like where they first like introduced ion
Hmm.
for enzyme. And then I guess I'm thinking of like the bonds that bring residues together. So they give like phosphodiester bonds,
Hmm.
which are typically between phosphates and
Hmm.
ox.
Yep.
Yep. Okay. So these questions, I'm like,
Hmm.
what? Like, I don't know what it is about these particular questions,
Yeah.
but they always get me.
Hmm.
Okay, so vDNA sequence encoding a protein is inserted into a host genome by IN. So basically, IN is doing its job right now.
Hmm.
It is then translated from the hypothetical mRNA sequence shown. So this is being, okay, so this is translating whatever IN brought in, essentially. So based on the passage, the segment of the original viral genome that encoded this protein had what nucleotide sequence? Okay. So I think typically, so I have a habit of like, again, going back to like the reading really quickly. And I think it comes down to like, I mean, that's kind of irrelevant. But basically my point is, I think if I were to read this over super quickly, I would like miss the fact that like, oh, the segment of the original viral genome. And so I think once I look at this, I'm like immediately thinking, okay, like what would this translate into? But that's not necessarily what it's asking. It's asking what, it's kind of like a trick question, to be honest.
Hmm.
Um, um, yeah, so I mean, so I guess another point is like this isn't making this the distinction that this isn't the sequence that's translated. This is what it's being translated from. And so if it's
Hmm.
being translated from something and it's asking what the original genome is, then it would be exactly this.
Yeah. Yeah. So it's like, like the codings, like the sense strand, aka like being the coding strand, and complementary to that would be the template strand.
Right.
And then complementary to the template strand would be that as equal the same thing as the coding strand, but
Right.
it'll be just complementary to the template strand, right? So yeah, so you have the right idea here. Yeah, but yeah, go on.
And so it should be A because nothing has changed. And they didn't mention like any mutations or anything.
Mm-hmm.
I know the viral um, mRNA would have like uracil. So,
Yeah, so this is what makes this question hard, in my opinion, because when I see DNA, I'm like, all right, no uracil, but we got HIV here, which is, you know, reverse transcriptase, right?
mhm,
Where DNA, or rather what like RNA would be translated into, or sorry, would be transcribed, reverse transcribed into DNA rather than DNA being transcribed into RNA. So that could make this hard. So, but yeah, what do you think it would be?
and I guess like something that I would remind myself too is the fact that like viral DNA can have DNA or RNA in it too.
Hmm.
So not being completely turned off by the idea that
Hmm.
there's self-different couldn't be the answer because it could very well.
Yeah. Yep.
Yeah, so
Yeah.
yeah, but I remember missing this the first time because I completely just um glossed over this part because I thought I was assuming that I knew what it was asking of me
Hmm.
and wrong. So yeah.
Yeah. That's a common thing. Um, like a lot of people who take the test, like they will kind of like try to get through the passage quickly, try to get through the question quickly, and then kind of look through the choices and frantically go back and forth from the passage to the choices and kind of play mix and match. But it really helps to slow down, and especially what can help to get into the practice of doing is to rephrase the question, because that'll
Mhm.
make sure that you understand what they're asking you. I think
Yeah.
it's very underrated to do that because, you know, because of like the time crunch, but certain things, if you spend time on it, it makes it worth it at the end. Yeah.
Yeah. You're right. Okay. So which experiment would best provide data to support the mechanism by which an ODN inhibits ion activity? Um, and so I guess for these questions, like you probably wouldn't predict an answer, right? You would just like kind of just go straight into looking at the options, or would you predict?
Let's try it. Because you could do it, like come up with your answer, right?
Mhm.
Because like, okay, like what are you thinking when you see this?
Um, so if I were to do an experiment that basically showed that like what they're saying in the passage is in fact like correct, I guess, like the same pathway is correct, then I would probably do something along the lines of like um like putting the enzyme in the presence or absence of uh,
Hmm.
like the viral, I guess.
So let's think about what comes to mind when you think of inhibitors. Like, um, like, yeah, what comes to mind?
So inhibitors will just inherently reduce like activation, so you'll see
Uh-huh.
less
So
activity.
like, think like what other things come to mind? Like, like for like one, so like for ODNs compared to legends, like we know that they're both inhibitors and they're both going to make the reaction not really work well.
Yeah,
But what are some like concrete kind of terms that we can use?
sure. So as it relates to like competitive inhibition that I mentioned in the last paragraph, so like I would I would expect to see like in the presence of more ODN,
So
the
presence and not an absence of the inhibitor, right?
yeah. So I guess the way where I was going with that was like the more concentration that ODN would be in this cell, it would have like a lower affinity
Mm-hmm.
for um
So like KM
yeah, KM would increase.
and like so what we would want to look for would be like changes in KM. What else?
Um,
Maybe not particularly for competitive, like it might be the same, but what are some other things that come up for inhibitors?
um
Maybe like Vmax.
okay, yeah. I mean, but if it's a competitive inhibitor, then we
Yeah,
wouldn't see
yeah.
yeah, feedbacks.
But generally speaking, like when we see the buzzword inhibitor, we want to be thinking KM, right? Vmax,
Yeah.
uh, substrate concentration, because KM is the substrate concentration at half Vmax, right?
Mhm.
So 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 yeah, we just want to be aware of like
Like what the trends should be, I guess, or is that
well
what you're
also like what like would comprise some type of way to figure out if a inhibitor is doing what it does. Like we would need to have the enzyme, have the substrate, have some way of measuring, how the how the enzyme converts the substrate into the product. And that can be its own thing. And then we can add an inhibitor and then measure those same things, right?
okay.
So that could be something that we do. So those are just things that I want to like have come up, like pop up in your brain. Um, yeah, and now we can kind of take a look. Yeah.
Okay. I think and to your point, I think like my initial thought process would just be like, oh, like what mostly aligns with like what we're expected to see in competitive inhibition, essentially.
Mm-hmm.
Okay, so the first option is keep ion concentration constant and measure the initial velocity of the reaction at increasing vDNA concentrations in the presence and absence of the ODN. Mm, I don't think it's immediately clear that I would mark that out. I think I would like hold on to it just to see like if others sound more
Mm-hmm.
fulfilling. B is keep vDNA concentration constant and measure the initial velocity of the reaction at increasing concentrations of ion in the presence and absence of the ODN. I guess one thing that I'm even thinking about is like well, I think we've already decided that like ODN should be the thing that we should like adjust the absence and um or presence rather. So I think maybe I would be leaning towards like answer choices that would align with that hypothesis at first. Um, and so I think I would probably like I'd probably mark out C and D just because they don't kind of align with that.
Okay.
Um, hmm. And then I guess it's just a matter of like, do we want to keep vDNA constant or IN constant? Mm. Hmm. Okay, well, so IN should be kept constant because it's the enzyme. And yeah, I mean, I think that's enough for me to even like,
Yeah, when we look at this, right, you get like your Vmax somewhere here and whatever, half of it or initial. But what would be on the x-axis?
a substrate. Yeah.
And what would be the substrate in this?
Um, that'd be would be ODN or VDN. No, no, no. It'll be VDNA.
Mm-hmm.
Yeah.
Yep. So yeah, so okay, you determine that ODN, if it's the inhibitor, we want to measure things with it and then without it.
Mhm.
So C and D don't work. And then it's a matter of what do we keep constant and then what do we have increasing concentrations of? And from this, we know that as we go to the right, we have increasing concentrations of the substrate.
Right.
And Michaelis-Menten, right, that assumptions involve like the enzyme concentration being constant and being much smaller than the substrate concentration, right?
Mhm.
So yeah. So yeah, so yeah, you would keep the enzyme concentration constant and increasing concentrations of the substrate, right?
Yeah.
Mm-hmm.
Okay. I think the first time I did this,
Mm-hmm.
I guess I just like wasn't really orienting myself. Like I wasn't reminding myself that like, okay, well, remember, this is
Mm-hmm.
the enzyme, like this is the
Yeah.
substrate. It's like
That's
I guess when you break it down that way, it's like, yeah, I know all of these things. It's just a matter of
mm-hmm.
like, I guess, I don't know, I guess like staying disciplined in that. Like I'm not going to
Mm-hmm.
uh I guess allow my biases to like,
Yeah, yeah. Yeah.
yeah.
That's why when you asked about like, should we just go at it? I was like, let's not, and then I just covered it. Because a lot of times, like, it depends obviously on the type of question. Like some questions you can't really answer without looking at it, but a lot of times you can, and that can enable you to be less susceptible to like wrong answer choices and have an idea, like a more clear idea of what to look for.
Mhm.
Um, but yeah, good job.
Okay, amino acid substitution for the conserved residue at position 64 is least likely to affect. So the way that I would reword this is like what wouldn't change, like the function kind of
Mhm.
based on quality. So position 64, so this is aspartic acid. So least likely is glutamate because they're
Mhm.
negatively charged. Yeah. Okay.
Perfect. Oh, and
And
we knew something like this was going to come up, right?
yeah, an inactive tetramer of iron is expected to have approximately what molecular weight? So one of the things that I have is 288. And then I remember it was, so it's basically 280. Well, okay. An inactive tetramer. I think like in my head, I'll think, okay, tetramer. So now it's like, would this be um like multiplied by 4? Okay. So you multiply 288 by 4 divided by 110. So it's about looking at A, I think.
So like walk me through like what you're writing.
So I would do I would round it 300 times 4. Okay, I didn't do my math wrong. So 300 times 4, 1200, and then dividing that by 110, which is the total Daltons in one amino acid. Uh, and then I know at this point, like
Mhm.
my answer's in the tens position. So it's would you rather just divide it by 100 instead of 110?
Yeah, yeah. So for the math stuff, right? Uh what I would do is like scientific notation. So like the 300 I'm going to just write as 3e2. I use e, that just means times 10 to the second power.
Yeah.
So like 3e2 um times 100, so 1e2 should give me something in Dalton. So this is going to be like the 3 times 1, so 3 times for the exponents here, if we're multiplying them, we're going to add them. So 3e2 times 1e2, 3e4. Oh, I forgot to multiply it by 4. So I'm going to multiply it by 4. So that's going to be 12 e4. And that would be the answer in Daltons. But since our choices are in kilodaltons, I'm just going to divide it by, or rather multiply it by 1,000.
So what's a D? It's
Yep.
D. Okay. Yeah, I think because I usually don't do my math that way.
Yeah,
I usually
yeah.
like what you do,
Mm-hmm.
except like do times 10 to like whatever power.
Mm-hmm.
I think, yeah, let me not try to do that. Okay. Because I think when I do it the other way, like because I'm rounding so much, I'm just like, okay,
Mm-hmm.
well, now I have now
Yeah.
I know what the
Yeah.
exponent is. And I realize
Mm-hmm.
that, like, oh, there's 3.
Yeah. Yeah.
Okay.
Mm-hmm.
Yeah, let me stick to oops. Okay. And then to determine if a small molecule acts like a legend with respect to ion, a researcher plans to incubate purified ion both with and without the small molecule, then perform a Western blot to detect ion in each sample. Under which conditions should the gel electrophoresis step be performed? Okay, so first thing I'm thinking about is like, okay, small molecules that act like legend. Okay, so basically these small molecules are equivalent to legend, like whatever legend does, these do. Um, and so now the researcher wants to incubate purified both with and without the small molecules. Okay, so essentially we want to know what happens when legend isn't present and when it isn't. Um, and legend is the inhibitor, so we should see less activity of ion when legend is present, and then the opposite when legend is not present. Is that correct?
Mm-hmm. And tell me like the significance of these types of gel electrophoresis.
Um, it's a way to see like whether or not um like mm, like substance is present and then like how much.
Uh, a little bit. Like, what would separate a native gel from like a denaturing gel, for instance?
Okay, so native. It's basically looking at like binding interactions.
Mm-hmm.
And then I know reducing is more so interested in disulfide bonds. And then I think denaturing just wipes out the co non-covalent interactions.
Mm-hmm. Yeah, right. So like when proteins, so like primary structure, secondary, tertiary, right? And quaternary. Um, so okay, like enzymes, well, no, proteins fold, and that lets us go to like secondary structure and tertiary structure. And denaturing is just unfolding. So it'll eliminate like tertiary structure and secondary structure. Um, unless it's disulfide bonds. If there's disulfide bonds to get rid of those, you would need reducing. So, so yeah, that's what to think about when you see the different gel types. Um, and yeah, you can try the rest.
Under which conditions should this gel electrophoresis be performed? Okay, so mm, mm, we definitely don't care for disulfide bonds because it doesn't seem like those
What
would
what do we care about?
um binding interactions because I think we're most interested in like its interaction with the active site in this enzyme.
So for the legend thing, what does that do?
So it inhibits it. So
A little, tell me more.
it keeps ion from having an active tetramer. So it basically wouldn't be able to like fold. Okay, there we go. Okay. So in that case, we're more interested in denaturing. Yeah.
Well, legend would need would make the tetramer, right?
Hmm.
So what type of gel would keep that tetramer as a tetramer? Because if you denature it or reduce it, you would break the bonds that would let it be a tetramer, right?
Hmm. And I suppose, I mean, those are also um, side chain interactions too.
Mm-hmm. Yeah, like so like secondary structure is going to be, well, primary structure will be the actual covalent peptide bonds, which are the amides, right? And then there could be hydrogen bonding between the backbone, which would give you the secondary structures of like alpha helices and beta pleated sheets. And then hydrogen bonding from the actual side chains, in addition to basically all the other stuff like electrostatic interactions, dipole interactions, but primarily hydrophobic interactions will be responsible for the tertiary structure.
Mhm.
So if the passage is telling us that legends cause, sure, it can inactivate it, but it still makes it like it's still going to be a tetramer.
Mm.
So what type of gel would work with a tetramer? Because certain types of gels would eliminate that tetramer, right? And give you like either the two dimers or the monomer, right? So we want to look for what type of gel would keep that tetramer as a tetramer.
That would be native.
Yeah.
Okay. So reducing is specifically disulfide bonds and then
Yeah, breaking them.
denaturing. Yeah. And then denaturing is whether or not like if we're concerned about like the activity, I guess.
Yeah, so yeah, denaturing will just cause unfolding through all of the types of intramolecular interactions except for disulfide bonds. And then
Mhm.
reducing would break those disulfide bonds. So, we don't know like for we don't know which specific type of bond can make IN turn into a dimer, and we don't know which specific type of bond would make it into a tetramer. But we
Mhm.
know that reducing and denaturing would likely break those types of bonds. So
Mhm.
if we wanted to keep that tetramer, like it's an inactive tetramer, but it's still a tetramer. If you wanted to keep that tetramer, we would need to run the native gel because the other types would likely break that tetramer up.
I see.
That's typically what you would see these gel questions like ask you about. Like
Mhm.
they could also ask you like how like like how many bands could you see, right? Like native would be like one band, and then like denaturing or reducing would be like two or four. So yeah, that's kind of what they're asking.
Okay.
Mhm.
So would you say, because I know that native is like very much binding interactions. Like that's kind of like the way that it's like encoded in
You say
MIME.
binding interactions? Tell me a little bit more.
Um, mm, when I say binding interact, okay, maybe I'm not even I'm not even saying that right. It's kind of like like it's default, I guess, structure almost is what
Hmm.
yeah. And
Hmm.
so if we want to analyze whether or not um like that default structure that they've defined that exists within this molecule, then we would want what we'd be more interested in.
Hmm. Yeah.
Okay. So then it would be three only because the other one would mess with that.
Hmm. Yep. In the chat, I put a picture. You can kind of share like that on your screen.
Can you see it?
I think you'd have to like because I think you're just sharing like the tab.
Oh, sure.
Yeah, there you go. So yeah, the primary structure is just that amino acid backbone through the dipeptide bonds, and there could be some disulfide bonds as well. That determines how a protein will fold. The secondary structure is the spatial arrangement of the amino acids near one another. So you'll have hydrogen bonds between the backbones. So we get the alpha helix here, side view, and the top view, you can see that the R groups are like outside of it. So the R groups are not playing a role in secondary structure. And then we can also have a beta pleated sheet. Same thing would be hydrogen bonding between the backbone. And then tertiary structure has a variety of things. Um, we get to have these electrostatic interactions here, disulfide here, hydrogen bonding between the R groups here, but it's predominantly going to be hydrophobic interactions that determine tertiary structure. And then quaternary structure is just if you have more than one subunit. Um, but but yeah, essentially, denaturing would break apart the secondary structure and tertiary structure,
Yeah.
and reducing would break apart disulfide bonds. So, and then native would just keep it the way it is. So,
Yeah.
that's,
Okay.
yep. So, yeah, how do you feel about how we went over this?
Yeah, this is super helpful. Yeah, I think practicing just kind of like orienting myself with the
Hmm.
past really. So I guess in your opinion, when you get through, let's say you're taking the MCAT and you got through like a passage that you didn't fully understand,
Mm-hmm.
how would you still work through that? Like not, because I mean, we did a pretty thorough
Mm-hmm.
breakdown of this
Mm-hmm.
that we kind of knew everything
Mm-hmm.
someone gave us. So yeah, if I wasn't able to do this on an exam, like how would you, or maybe not necessarily if I wasn't able to, but like not as thorough, I guess. Maybe they're like
Yeah,
safe.
yeah. Like, so like I guess like yeah, how to translate this into like what could be something that you use like in a week. Um, well, okay. I guess like I should maybe first tell you that like a timing kind of strategy that could help. Like let's say you get to question 20, I would set up like checkpoints for timing. So like once you get to question 20, uh check the clock. You should have like an hour left if it's a science section. And this gives you like instead of constantly looking at the clock, we're gonna have three checkpoints where we will look at the clock. And at these three checkpoints, we can adjust our pace. So when you get to question 20, check the clock and see if you have like an hour left. If you have more than an hour left, you're good. If you have less, go a little faster. Next checkpoint will be question 30. You should have 45 minutes left. And last checkpoint, question, oops, 40. You should have 30 minutes left. So this will just give you three checkpoints at which you can change your pace. So that's just general timing strategy to maybe help you, with the timing and everything like that. But yeah, for the passages, like, when you're taking the test, yeah, it's hard for me to kind of encapsulate that into like, you know, into like what to do, I guess.
I mean, maybe my priority should just be like practicing and like,
Yeah, like,
yeah.
because yeah, so, you know, well, okay, so I guess you can see that from this passage that it may not, well, okay, so well you saw that certain buzzwords mean that they'll likely ask you questions about it, and we kind of saw that coming in some ways, but also in other ways when things may not readily be apparent, like how it's like for this question that they're asking about these gels, and it may not be readily apparent like why they're asking about the gels, but if you know that the gels are there to ask you kind of questions about like fragments that you'll see, that can kind of help because they'll likely not make it so, I guess, complicated if you kind of understand like the basic ideas of the things that they'll ask you about from when you read it. But let's see. Um, yeah, I guess I would say like as you read the passage, um, when you see certain buzzwords that you should be thinking of certain things that they'll likely ask you. And if you don't understand something like right away, like for this passage, I think there was this part about the there's something about some dinucleotide or something. Oh, this part,
Hmm.
IN catalyzes cleavage of a GT dinucleotide from
Hmm.
each 3' V DNA terminus. We kind of like brushed over that because it could possibly be something that if we try to like understand it could make it so complicated that it would work against us. So we kind of maintained like a big picture view when we kind of visualized like the ends of it with the dimers and then they come together. You were able to say that. Um, so So I guess, you work with, I guess, deciphering everything into what you already know, and I guess understand that most of the time you can break things down into things that you already know and then kind of go from there. So things that might feel like when you're reading it that you don't know that much about likely is something that you do know a good deal amount. Um, but it's just, I guess, through practice, you're going to get used to being able to, like, when you see a certain type of topic, to be like, this is what they'll likely ask you about. Um, so like for this one, it was like enzymes, active site contain certain amino acids, and they asked us about that, and they asked, like, what's an amino acid that would maintain the function and what, wouldn't maintain the function, the amount of residues, like when we saw that with the 288 residue protein, but when we saw the dimer and tetramer, they'll likely ask you about the weight, but for like the dimer or tetramer, to make it so it's not completely just like one step kind of thing. Um, I'm not really sure. I'm sorry. Like, uh,
I think that's
like
valuable.
like what you read, you just want to like put it into the category of this is something that I can figure out that isn't completely out of left field, even though it might seem like it
Mhm.
is maybe the best way I could put it. So, you know, so for, most of this stuff, like it wasn't really any new stuff, like new content, like, right? It's just like you're kind of fitting it in with the content that you already know, and you
Yeah.
just kind of go from there. So, yeah, I would say, and like, yeah, like get into the habit of like visualizing, I guess. It could be like really simple, just things with circles. That's kind of like what I'm doing. Um, yeah, I don't know.
Yeah, no, I think that's super useful because I haven't been doing enough of that, I think.
Yeah, yeah.
And like what's been holding me back from scoring
hmm
a lot. Yeah.
Yeah.
Yeah. And a lot of this also comes with just like experience, just like keeping
hmm
practice. So yeah, gonna
Yep.
continue.
Yeah.
Yeah.
Yeah. I would say for like the full lengths that you do, like when you like do them, do something called like a blind review. What that means is, like, let's say for CP you get 10 questions wrong, like write down which 10 questions you got wrong. Try not to look at the answer, and then just open it like with the Jack Weston Chrome extension to hide the answers, and just try these 10 questions again, but this time without the time constraint.
Mhm.
So this lets you separate the questions you got wrong because you really didn't understand them from the questions that you got wrong because you're out of time.
Yeah.
So this will mean that, like, you're going to be working through some of these problems, and you'll likely get some of them right when you do it, without the time condition. So let's say after your blind review, now you just have 5 wrong. Then you can do your regular review, which is like looking at the solutions. And
Mhm.
let's say like 2 of them still don't make sense. When we meet, we can go over those 2. Or if you did understand something, but you just you're curious about how I would go about it, we can also spend our sessions doing that. But I think doing a blind review will make the most out of the days that you have like left, and the tests that you're going to be doing in these last like 2 weeks.
Yeah. Yeah.
So
Okay.
like like if you can just like do like problems and minimize problems that you don't like redo. No. So like, let me rephrase that. If you can maximize like blind review type of thing where you're actively working through problems, I think that you're gonna have a lot of these things kind of autocorrect,
Mhm.
compared to if you kind of like do it more passively with reading the solutions.
Yeah.
Um, so I think the blind review will help you out with, kind of a lot of this stuff. Yeah.
Okay. Yeah. I'm gonna do that for my next practice. then.
Cool.
Yeah, sounds good then.
Mm-hmm.
All right.
Mm-hmm. Awesome. So do you want me to put you in for next time, or do you want, like, what do you want to do?
Yeah, so I'm gonna take my practice tomorrow, and I'm
Mm-hmm.
gonna do the reviewing that you mentioned, and
Mm-hmm.
then um we can go over like things that I guess aren't autocorrecting, I suppose is the way you put it.
Yep.
Yeah. Yeah, and I think I take pretty long when I review my
Mm-hmm.
practice. So I don't even, I mean, part of me is like, I mean, it's all kind of useful time, but then
Mm-hmm.
it's like, I don't know. I feel like maybe I take too long really like sitting there and digesting. Because I do have a habit of like redoing questions just to see like, oh, will I get
Yeah.
it this time around? Um, but that also tends to take like more than one day
Yeah.
at least. So
Um, yeah, okay. Let's see what we can do to make it. When you say more than one day, like 2 days or like 3 to 5 or something. Like,
say 2 days.
okay.
Yeah.
Yeah, I don't think that's too bad, especially if you're gonna do the blind review and stuff.
Hmm.
So yeah, maybe we can try to make it like one day or something, but it doesn't sound too bad.
Okay. Sounds good then.
Hmm.
Maybe we can schedule another time for Wednesday. I think Wednesday would probably be
Let's
if
see.
you yeah, if you have any
What would 3 p.m. Eastern on Wednesday work?
yeah, that'll work.
Okay.
Yeah.
All right. So I put you in for, yeah, Wednesday the 19th at 3 p.m. Eastern, okay?
Okay. Sounds good.
And do you have Venmo or Zelle or something like that?
Yeah, so I can do either, to be
Okay.
honest. What do
Yeah,
you
Zelle would work. I will put my phone number in the chat.
gotcha.
And let's see. This is okay. So let's see.
And you said Zell specifically.
Mm-hmm. Okay. This will be 96 dollars. I did the 75 per hour thing, and then I took 15% off of that for
Gotcha.
1.5 hours. So yeah, 96.
Gotcha.
Mhm.
Okay, okay, put in your number. All right, got it. And then I'll set it right now, and then you can let me know. Or would you be able to know? We got it.
Uh, I'm also sharing a Google Drive with you that has a lot of useful stuff in it.
Okay.
Sorry, what were you saying?
I was saying I'm gonna set it now, then you can let me know if you
Hmm.
get
Yeah.
I think you're still recording too, in case you didn't know.